The Nernst equation is an equation that predicts the equilibrium potential for a given ion based on the concentrations of the ion on opposite sides of a permeable membrane. It also predicts the voltage difference needed to counterbalance the diffusion force. The Goldman equation is an equation predicting the potential difference across a membrane based on the concentration of ions on opposite sides of the membrane, as well as its relative permeability to each ion. It includes the intracellular and extracellular concentrations of K+ and Na+, and the degree of permeability. It also predicts a voltage potential that is similar to a neuron's resting potential. Inside the neuron, there is a high concentration of potassium. Outside the neuron, there is a high concentration of sodium. A threshold amount of depolarization triggers an action potential. Action potential is a short and big change in polarization within the neuron that is propagated quickly along the axon. These action potentials are responsible for the transmission of information between neurons and their target cells. Hyperpolarization is the inside of the cell becomes more negative relative to the outside. Depolarization is the inside of the cell becomes less negative relative to the outside. Capacitance is the membrane's ability to store electrical charge. Graded response is a neuron's change in potential. Local potential is an electrical potential that is initiated by stimulation at a specific site, which is a graded response that spreads passively across the cell membrane, decreasing in strength with time and distance. The threshold is a stimulus that triggers an action potential. Larger depolarizations produce more action potentials, not larger action potentials. The all-or-none property is the size of the action potential has no effect on the size of the stimulus. If the action potential does not fire at full amplitude, it does not fire at all. Changes in frequency are more important with regards to action potentials compared to amplitude. After potentials are a change in membrane potential after an action potential. It could be negative or positive, and it is due to the movement of ions into and out of the cell. Ion mechanisms underlie the action potential. English neurologists Alan Hodgkin and Andrew Huxley studied the axon of a squid. This neuron was involved in the squid's emergency escape behavior. It was observable by the naked eye, more than half a millimeter in diameter. They manipulated the concentration of ions that went in and out of the axon while recording the electrical current inside the membrane. These neurologists discovered that action potential is created with the help of voltage-gated Na+ channels. Voltage-gated sodium channel is a selective channel for sodium ions that open and close in response to electrical differences in membrane potential. Only the first stimulus can evoke an action potential. The second stimulus triggers a refractory period. Refractory period is an exhaust axonal membrane. There are two refractory phases. There is an absolute refractory phase, which indicates complete insensitivity to stimuli. So that means that no amount of stimulation can trigger another action potential. The second one is a relative refractory phase, which is a period of reduced sensitivity, which means that only very strong stimulation can trigger another action potential. The transfer of action potentials is found only in the axon. Action potentials are actively propagated along the axon. The axon potential travels down the axon at a rapid speed. Action potential is renewed as it travels down the axon. Action potentials only travel in one direction, from the axon hillock to axon terminals. This is because action potentials leave refractory membrane behind after the potentials occur. Conduction velocity is the speed at which an axon potential travels down the axon. This varies with diameter. Larger axons are faster than smaller ones, and the myelin sheathing also adds speed. A node of Ranvier is a gap between successive segments of myelin sheath. Action potentials jump from node to node, and this is known as saltatory conduction. This is an evolutionary advantage that humans have. Voltage clamping and patch clamping. Voltage clamping is the use of electrodes to inject current into an axon or neuron to keep the membrane potential at a set value. The apparatus measures how much current must be injected to counteract any ion channel openings. This technique was used in Hodgkin and Huxley's squid study with the squid axon. One electrode is placed inside the axon and another in a bath surrounding the axon. Scientists discovered that this apparatus could be applied to neurons and cells as well. Patch clamp is a use of voltage clamping to monitor current flow across a tiny patch of membrane taken from a neuron. Changing the channel. The cell membrane is made up of fatty molecules that repel water. Selective ion channels in the membrane aid in the transport of certain ions. Channelopathies are medical conditions that arise from abnormalities in the form and function of ion channels. Channelopathies are due to gene mutation. For example, a sodium channelopathy is related to seizure disorders, muscle disorders, and cardiac disease. Chloride channel disorders are related to deafness, kidney issues, and movement disorders. Certain animal toxins can block the voltage-gated channels, sodium channels. For example, tetrodotoxin and saxitoxin are two examples of these toxins that block voltage-gated channels. Even though these toxins can be deadly in certain cases, they can also be helpful in others. They can show how the toxins block the channels, and they can be used as local anesthetics as well.